Welcome back. So we're going to get into some more details about meiosis today. So I just wanted to quickly review what this looks like. Remember, this is going to take place after the S phase, after the DNA has been replicated, and the cytoplasm has also all been growing so that it can prepare to divide this cell. And remember, these are our germ cells. So we're starting off with a diploid germ cell, and throughout the two phases of meiosis, the first phase of meiosis, we are going to be pulling apart the homologous pairs, and at the end of meiosis 1, we're going to have two haploid cells. And then each of those haploid cells is going to go through meiosis 2, and we said that's pretty similar to mitosis in each of those phases. And at the end, we are going to wind up with 4 haploid cells. Oh, that's a terrible N. How about that? We'll make it a capital N instead. That way. Oh, wow. Y'all, I am terrible at writing with this pen. Anyway, at the end, we have 4 distinct genetic haploid cells. Okay, so let's start with meiosis 1. Remember that our parent cell is diploid, and it has just gone through the S phase. And in the S phase, the homologous chromosomes basically replicate, and they undergo a synapsis, which results in what we call a bivalent. And so there's a picture of that in this inset here. So it's basically 4 sister chromatids or a pair of homologous chromosomes. And I just wanted to make sure that you understood the definitions. When we talk about homologous chromosomes, or sometimes they're called homologs, that's when each chromosome or each chromatid has the same genes in the same locations, and that enables the pairs of chromosomes to align correctly with each other before separating during meiosis. Okay, so I went ahead and just wrote that out so you'll have that definition. So during prophase 1, which is this first step here, we know that nuclear division is about to occur, so the centrosomes begin to move away from each other. Remember, they pair together, and they start to migrate to either end of the cell. So they start that process. And the nuclear envelope begins to break down, and the nucleolus disappears. That's prophase 1. Then we move on to metaphase 1. In metaphase 1, these bivalents are going to move towards that metaphase plate in the middle, so that imaginary line that looks kind of like an equator of the cell. And when they do this, each bivalent is aligning independently of the others. And that's going to contribute to our genetic diversity. We'll talk about that in just a minute. But they will be oriented toward either pole of the cell. So it's getting ready to split. And then we go into anaphase 1. And this is when the homologs of each bivalent separate. So they're being pulled apart towards either end of the cell based on those spindle fibers. Just keeping in mind that the sister chromatids don't separate, but each bivalent pair is separated. So this is when we go, we're starting to split it from diploid to haploid. Then we're going to enter telophase 1. And this is very similar to telophase in mitosis. And that is like the spindle fibers start to disappear, new nuclear envelopes are being developed. This is where all that splitting actually occurs. And depending on the cells, this may or may not be accompanied by cytokinesis. In other words, the splitting of the cytoplasm isn't as important, so they may look a little different. And so at the end of this, we wind up with 2 haploid cells. Then we enter what we call interkinesis. And so what that represents sort of is like kind of like the interphase portion of mitosis, where it was preparing for the next division, just sort of that resting period, except for that right now we're not going to duplicate the DNA again. So kind of review those steps in meiosis 1, and we're going to prepare for meiosis 2. Okay, so here we are setting up for mitosis 2. So this is the second set of phases of meiosis. So we begin with our cells from meiosis 1, and the cells have 1 chromosome from each homologous pair, so they are haploid. And so in prophase 2, we're going to see, just like we have in all the other prophases, our spindle fibers are starting to develop. There's a breakdown of the nuclear membrane, just like we've had before. And then we're going to move on to metaphase 2. And during metaphase 2, we have an alignment of chromosomes at the plate. And note that the chromosomes still consist of duplicated sister chromatids at this point. So they're aligning in homologous pairs just like they do in meiosis 1. But because only 1 chromosome of each homologous pair is present, that's what you're seeing line up. Okay, then we're going to move into anaphase 2. During anaphase 2, we have a separation, and this time it's a separation of the sister chromatids. And these daughter chromatids are going to be pulled to the poles from the spindle fibers. Then we move into telophase 2, and this is where we have that separation. We have telophase 2 as well as cytokinesis. And so the spindle fibers disappear, the nucleolus starts to form with that nuclear envelope again, separation of, or division of the cytoplasm, and then we have the resultant 4 daughter cells that are all haploid. And I know that you can tell in all of these diagrams that the colors of the chromosomes and the chromatids make a difference, and we're going to talk about that in a minute. Right now I just want you to see the process of how we're getting to 4 daughter cells that are all haploid. So in animal cells, these daughter cells are going to be your gametes, so your sperm and egg. In plants it's a little bit different, because these plants, these particular haploid cells are then going to undergo mitosis and form a multicellular organism that's all haploid, and that's the gametophyte generation. And from one particular part of that multicellular organism, they're going to produce sperm and egg. But because plants have part of their life cycle where the multicellular organism is all haploid, we call that alternation of generation. So whereas animal cells like us spend almost all of our life in the sporophyte generation and just have the sperm and egg as the gametophyte generation, plants are different because they are multicellular organisms that go back and forth between haploid and diploid phases. So that's just a little bit different. But it's fine with me if right now we're focusing on meiosis 2 produces gametes in animal cells. So how can 2 parents produce so many different types of offspring? The answer is that all sperm and all eggs are genetically different, and we get that through meiosis. So some of the things that we didn't talk about within meiosis is what happens with all those chromosomes and how do we get diversity within the genes. So let's talk about that. One of the ways to achieve this genetic variation is through crossing over. So crossing over occurs during prophase 1 of meiosis 1. And what that means is it's an exchange of genetic material between non-sister chromatids within a bivalent. So if you remember, during prophase, homologous chromosomes come together in pairs to form a bivalent, or sometimes that's called a tetrad because there are four chromatids all together. And so what happens is during that crossover, that genetic material is exchanged when that bivalent is put together, and we're going to look at that in a little bit more detail. But this happens usually in humans about 2 to 3 times per meiosis. So if you think about that, that helps to give us some genetic diversity. So let's talk about what that looks like. Okay, so during prophase 1, at what we call synapsis, which is basically the pairing of homologs, the homologs line up side by side, and then this nucleoprotein lattice appears, and you can see that in this electron micrograph right here. So it's a temporary holding together of those homologs. And so that forms what we call the bivalent that we were talking about. Remember that bivalent has all 4 of these chromatids. So during crossover, well, let me back up for a minute. While we align these chromatids, they're aligned in such a way that all the genes, all the locations or loci of each gene aligns for each chromatid. And so it's at that point at which DNA material can be exchanged between these chromatids, or what we call crossing over. And so if you'll remember, the homologous pairs actually contain different alleles for the same gene. So once we take that into account and consider that we have now exchanged DNA between chromatids, we have a lot of variation that's being generated. So once that crossover occurs, the lattice breaks down, but there is still what we call a chiasma. The chiasma is basically the holding together, because the DNA is still holding on to each other, of non-sister chromatids. So in this example, we have exchanged information between chromatid 1 and chromatid 3, and so that chiasma is there temporarily. Now, because of this crossing over, the chromatids that are held together by the centromere are no longer identical like they were before. So when the chromatids separate during mitosis 2, then the daughter cells will receive DNA that's been recombined. So we call that genetic recombination. And this genetic recombination is important because that's where you get a whole variety of genetics and of different alleles. And so the daughter cells and the resulting organisms from them, once we have fertilization, means that those offspring don't have the exact same genetic material as the parent, but have a variation from them. I don't know about you, but it makes a whole lot more sense to me when I can actually see this occurring. So here's a link to a video so you can just see what that looks like in video form. The second way that we increase genetic recombination or increase the diversity is through what we call independent assortment. And this happens during metaphase 1. So the bivalents, or the homologous chromosomes, separate randomly or independently from one another. So when the homologs align on the metaphase plate, the ones that either come from your mother or from your father can be oriented to either pole. So therefore, when they are pulled apart, it's random whether you get a particular one from your mom or a particular one from your dad. So in the example that we have here, this example only has 3 chromosomes. And so mathematically, when we're looking at the potential different combinations of those in the outcome in the daughter cells, we would say 2 to the 3rd power, so you have 8 different combinations. And that 3 is because we have 3 chromosomes, 3 homologs. Well, in humans, we have 23 of those, so if you take that then and do the math, you would have 2 to the 23rd, which gives you 8.4 million different combination possibilities. So you see how this in and of itself helps to increase the variety. So unless you're talking about identical twins, if you look around, nobody looks the same. And so between our prophase, where we have crossing over, and then in metaphase, where we have the independent assortment, we have quite a bit of genetic diversity within the population. And both of these take place during Meiosis I. So in humans, and in most animals, meiosis is used only for the production of gametes. And so the terms for that are spermatogenesis, which is the making of sperm, and oogenesis, which is the making of an egg. And so interestingly enough, when spermatogenesis occurs, you might be able to see in this diagram here, that it starts out with a primary spermatocyte, which again is your somatic cell, diploid cell, and it goes through your meiosis I, meiosis II, and you come up with 4 haploid sperm with all different kinds of DNA based on crossing over and independent assortment. For oogenesis, it's a little bit different. So you begin with your primary oocyte, which is again your somatic cell that's diploid, and you wind up producing 1 egg cell and what we call polar bodies. And each of these different stages of meiosis has some checkpoints. And in fact, when there is sperm present, the penetration of the sperm actually causes the last step of meiosis to occur. So that's kind of interesting. But those polar bodies that I talked about are in some ways a way to just get rid of the rest of the genetic material, and then, but in the meantime, it has allowed a lot of cytoplasm to be saved. And so the oocyte, or the production of the egg, is a much larger cell because the polar bodies contribute their cytoplasm, and so therefore you're left with just 1 copy of that genetic material. The others are basically trashed. And so from 1 primary oocyte, we wind up with 1 haploid egg. Whereas in spermatogenesis, we started with 1 diploid spermatocyte and wind up with 4 haploid sperm. But meiosis basically in humans is meant to accomplish the production of sperm and egg. So when everything goes well in spermatogenesis or oogenesis, we call this euploidy. And that means that the egg or the sperm cells that are resulting from those meiosis processes have the correct number of chromosomes. So they should each have 23. Then what happens if things don't go as planned? We call that aneuploidy. And that means that either the sperm or the egg results in an incorrect number of chromosomes. And oftentimes that happens because somewhere in meiosis something didn't split correctly. And so you wind up with either extra chromosomes or not enough. And if that occurs in our autosomal chromosomes, in other words our non-sex chromosomes, that generally is lethal. So either the egg dies, the sperm dies, or after fertilization, the zygote dies pretty quickly. So oftentimes women will have miscarriages for this reason. And in fact, a lot of women have miscarriages before they even know they're pregnant, because it does cause death very early. But sometimes there is survival, and it's very rare, but on chromosome 13, 18, and 21, when that happens, and typically that means we have an extra chromosome in what we call trisomy or a trisomic condition, those can actually survive. And on chromosome 13 and 18, a lot of times these children don't live past childhood. But chromosome 21 is, if we have an extra chromosome for chromosome 21, that is what we call Down syndrome. So there's lots of different syndromes that I could talk about. We're not going to go into all of those. I just wanted to bring this to your attention. But I also want to make sure that I in no way say that there's something wrong with any children that have aneuploidy. Just because they don't have the same number of chromosomes as everybody else doesn't matter. They have grown, survived, done great, and contribute in lots of ways to society. I have several friends that have Down syndrome children, and they are the most loving children. So I want to be careful how I word this, really. But anyway, I just wanted to mention that meiosis doesn't always go in the way that we have stated it here. This is the ideal. Sometimes it doesn't go exactly as planned. So now is a good time to just stop and think about these questions. I'm not going to answer them for you because I'm pretty sure by now through this lecture you should be able to answer them. But think about what cells undergo mitosis, what's the end result of mitosis, where in the human life cycle do you find mitosis, and in juxtaposition to that, why do cells undergo meiosis, and what's the end result of meiosis? And then what two phases of meiosis lead to genetic diversity? So here is a side-by-side comparison of mitosis versus meiosis. And so both of them are going to begin with a diploid cell. In this particular example, it has 4 chromosomes. And so we look at prophase in particular, shows a big difference between the two. In the case of meiosis, we form the tetrad, or what we call the bivalent. That does not occur in mitosis, but only in meiosis. So here we have chromosomal duplication, but we have the formation of the bivalent in meiosis. As we move into metaphase 1 and metaphase of mitosis, we have the bivalents are aligning on the metaphase plate in meiosis, whereas in mitosis, the chromosomes align on the metaphase plate. Then we move into anaphase and telophase, and that's fairly similar. We are, the only difference is that we are separating the homologous chromosomes in anaphase in meiosis, whereas in mitosis we are separating sister chromatids. And then we have, at the end of anaphase and telophase, in mitosis we have 2 daughter cells, both are diploid. And then in meiosis we continue on to a second division, which is going to leave us with 4 daughter cells, all haploid. Okay, so that is the end of meiosis and sexual reproduction. Take your time to review that and look at the summary and the differences of meiosis and mitosis. Make sure that you understand what phase comes when and what they accomplish.